Hi, my name is Noelle Mason, and I am a third year undergraduate in Dr. Kristen Ruick's lab. I'm majoring in fish, wildlife, and conservation biology, and today I'm going to be sharing my research on comparing stress in niche tracking and niche switching in a migratory bird, which is a yellow warbler in this case. So recent work has supported the fact that avian populations can persist in the face of climate change based on their extent of climate specialization across migration. So in 2021, Dr. Rachel Bay and Dr. Kristen Ruick put out a paper that quantified the extent of genetic substructure across the yellow warbler system, across the breeding ground. So they were able to find that there's distinct genetic substructure based on the environments that the birds live in on the breeding ground. They were able to find that birds in this red area on the top second to top left figure, the map there, those birds are on the breeding ground, and then when they fly down to the wintering ground, some of them go from a dry environment on the breeding ground to a dry environment on the wintering ground. But other birds fly from a dry environment on the breeding ground to a wet environment on the wintering ground. And the goal of my research is to figure out which of these two groups are more stressed. So the group that's going from a dry place to a dry place is known as niche trackers, and the group going from a dry place to a wet place is known as a niche switcher. So I'm trying to figure out if niche trackers or niche switchers are more stressed out. And I plan to do this by measuring relative telomere length in yellow warblers. So telomeres are, I like to think of them as the little plastic bit on the end of a shoelace that protects your chromosomes as your cells divide, and they can shorten, they naturally shorten throughout an organism's lifespan. But they can be accelerated in the shortening process by environmental stress. So the goal of my project is to understand if niche tracking or niche switching behaviors make telomeres shorten faster, and therefore understand which group experiences more stress over the course of their lifetime. And I predict that telomeres will shorten faster in niche switchers because they're experiencing more stress due to a higher variability in the environment over the course of their life. So I plan to measure telomeres using a method called quantitative polymerase chain reaction, and then analyze the data that I get about relative telomere lengths using linear regression. That will allow me to understand the stress impacts of niche switching and niche tracking on migratory birds. This may also help to illuminate the challenges that the yellow warbler and other similar migratory species are facing, either on their breeding grounds or outside their breeding grounds, depending on the results that I find out.